Namaskar dosto, welcome to channel Toxicology on Board. Today in this video we'll discuss another DABT topic that is in vivo micronucleus assay. So let's directly dive into the question. So question what we have is the in vivo micronucleus test is often used in standard battery of genetic toxicity assays. In this test, what do the micronuclei actually represent? So we need to identify what is this micronuclei, okay, the characteristic or the nature of the this micronuclei. So we have option A, clumps of ribosomes aggregated around rough endoplasmic reticulum. Option B, we have binucleated remnants of germ cells in the intracellular space. Option C, we have membrane bound structures that contain chromosomal fragments or whole chromosomes. Option D, we have pycnotic nuclei from a cell with decreased cytoplasmic to nuclear ratios and thickened micronuclear membrane. The right answer will be in green text, that is option C, membrane bound structures that contain chromosomal fragments or whole chromosome. Okay, so let us understand what is the explanation and the reference which is given by the American Board of Toxicology. So we have, because the micronuclei represent a chromosomal fragment, they are simple indicators of DNA damage, right? Both clastogenesis and aneuploidy. So you can identify both clastogenic damage as well as aneuploidy can be identified by this micronuclei. These micronuclei are not nuclear fragments or ribosomal clumps. Okay, the micronucleus test is run in lymphocytes or erythrocytes, not germ cells. Right? The test is this micronucleus test is widely recognized as one of the most useful and reliable tests for carcinogens that act by causing DNA damage. So we know that carcinogens we have two different mechanisms. One which follows DNA dependent and non-DNA dependent, in which molecular mechanisms or pathways are interrupted. One which in which the chemical agent will directly bind to the DNA and cause the damage. Such type of carcinogens, genotoxic carcinogens, non-genotoxic carcinogens. To identify genotoxic carcinogens, this particular micronucleus assay is a very suitable assay. Okay, reliable tester. The test is this micronucleus test is a part of the OECD guidelines for testing chemical safety. So we have a battery of genotoxicology test as recommended by the OECD. So in that one of the test is micronucleus assay. So what is the nature of this micronuclei? Once again, these are nothing but membrane-bound structures that contain chromosomal fragments or whole chromosomes. Certain times whole chromosomes, or certain times it will be chromosomal fragments. So let's see what else we can learn from this concept. So this is the reference which is given by the American Board of Toxicology, Casseret and Dowell, ninth edition, page number 525 and 526. So please read genetic toxicology chapter in the case of laser. So we have metaphase analysis. So in the case of cell division, you have two different prophase, metaphase, anaphase, telophase. In that metaphase analysis is there, no? It is a time consuming and requires considerable skill. So simpler cytogenic assay have been developed, of which micronucleus assay have become the most especially important to identify such type of damage. These micronuclei are nothing but similarly seen in the previous slide, no, membrane bound structures that contain chromosomal fragments or sometimes whole chromosomes that are not incorporated in the daughter nuclei. Okay, these are the micronuclei. Since micronuclei usually represents acentric chromosomal fragments, these are most commonly used as simple indicators of this chromosomal damage. Okay, acentric based on the where the your telomere centromere that will be there. No, that is called acentric chromosome. However, the ability to detect micronuclei containing whole chromosome has led to their use for detecting aneuploidy as well. So wherever we are identifying complete chromosomal is there we are able to identify aneuploidy. Micronucleus assay may be conducted in primary cultures of human lymphocytes. Test system might be, or it can be done in mammalian cell lines, or it can be done in mammals in vivo. So these are the three different in which cultures you can perform this micronucleus assay. So this is a figure which is taken from Clayson textbook only. Figure is 9.8, in which you can see this is a micronucleus in a human lymphocyte. The cytosalicin B method was used to inhibit cytokinesis. We have to block that particular phase in the cell cycle. That resulted in binucleus. So we have two different nuclei are formed. The micronucleus arrow, this micronucleus arrow is there, no, resulted from failure of an acentric chromosome fragment or a whole chromosome being included in this particular daughter nuclei following cell division. So let's see what else we can learn from this concept. In continuation only, which is given in page 525 and 526, micronucleus assay in lymphocytes have been greatly improved by cytokinesis block technique. How we are doing this one block by using, in which a cell division is inhibited by using this particular chemical, that is nothing but cytochalasin B, resulting in a binucleated or multinucleated cells. Okay, in the cytokinesis block assay, human lymphocytes non-dividing zero cells. We have G phase, S phase, G1 phase like that. So in that particularly G0 cells phase cells are treated with either with ionizing radiation or radioactive radiomimetic chemical, and then stimulated to divide with the mitogen phytohemoglutinin. Alternatively, the lymphocytes may be exposed to the mitogen first, so that the subsequent mitogenic treatment with radiation or chemical includes S period of the cell cycle. Okay, so cytochalasin B is added to inhibit this actin filament network formation and cytoplasmic division along with the test material when treating the cells continuously for 1.5 to 2 cell cycles in the absence of S9 fraction. For short duration exposure, for example 2 to 4 hours, cytochalasin B is added at the end of the treatment, and the cultures are harvested also after 1.5 to 2 cell cycles. In both cases, micronuclei are counted only in the binucleated cells, okay, so as to ensure that the cells have undergone a single nuclear division that is essential for micronucleus development. The assay avoids confusion owing to differences in cellular proliferation kinetics. Okay, so let's see what else we can understand about this concept. Although the first device used primary lymphocytes, the cytokinesis block micronucleus assay has since been adapted for use in continuous cell culture, including the Chinese hamster, human TK6, mouse lymphoma cells that are widely used in other genotoxic assays can also be done. So micronucleus assay should be considered in such a way that cellular proliferation is monitored along with the micronucleus frequency, and this is facilitated by this cytokinesis block by this cytochalasin B. Okay, so reliable data have been obtained in culture cells both with and without cytokinesis block, but scoring results only in the binucleated cells. After blockage of cytokinesis with cytochalasin B confers advantages with respect to the measurement of proliferation, recognizing whether an agent is cytostatic, okay, or obtaining clear dose response relationship. So this is what happens in the case of it is a double strand break is happening, formation of this micronucleus is happening whenever this binucleated cells is there. Along with the daughter nuclei, a small micronuclear fragment, either complete chromosome or part of the chromosome, will remain the daughter nuclei. So, although micronuclear resulting from the chromosome breakage compromise comprise the principal endpoint in the cytokinesis block micronucleus assay, the method can also provide evidence of aneuploidy, chromosomal rearrangements that form micronucleoplasmic ridges, inhibition of cell division, necrosis, apoptosis, excision, repairable lesions. So all these type of work can be identified by using this micronuclei. Micronuclei in a binucleated human lymphocytes are that particular picture I showed that was there in figure 9.8. Okay. Then a recent review of the International Human Micronucleus HUMN project provides a comprehensive description of discusses about the standardized protocols for this micronucleus assay in human lymphocytes and buccal cells. Okay, together with a review of association between this micronuclear data and disease outcomes. So these are the different steps are there. This is you can see very clearly the micronuclear which is induced by this particular genotoxic agent. Okay, and this is the cell daughter nuclei without micronuclei. Here in this daughter nuclei where you can clearly see formation of this micronuclei. Okay. In addition, we'll also see that the in vivo micronucleus assay is often performed by counting micronuclei in the immature, that is polychromatic erythrocyte PCE, in the bone marrow of treated mice, but it may also be based on peripheral blood. Certain times you will do peripheral blood micronucleus assay, and you can also do bone marrow micronucleus assay. Micronuclei remain in the cell when the nucleus is extruded out in the maturation of erythroblasts. When the final erythroblast cells are formed, no, this micronuclei come out of that cell. It will be extruded in the maturation in the maturation of this erythroblast, right? Whenever this division is cell division, it is taking place. In vivo micronuclear assays are increasingly used in genotoxic test as a substitute for the bone marrow metachromosome analysis. Okay, since we are able to identify all types of damage in the case of micronucleus, okay, it is a good replacement for other type of assays. Micronucleus assay have been developed for mammalian tissue other than bone marrow and blood, including skin, duodenum, colon, liver, lung, spleen, testis, bladder, buccal mucosal cells, stomach, vagina, and fetal tissues. Although assays in the bone marrow and blood are the mainstay of genotoxic testing, the new assays are important for mechanistic testing to understand these mechanistic studies and the research on the site specificity of the genetic damage and carcinogenesis. Getting my point? So these micronuclei are most commonly visualized through this microscopy, okay, by using this flow cytometry. But automated means of detecting micronuclei have been developed through application of this flow cytometry. So like this, you can do flow cytometric detection is effective in micronucleus assay in rodent bone marrow or blood. It can also be used to detect micronuclei in Chinese hamster ovary cells, okay, where altered flow cytometric parameters can reveal whether micronuclei errors primarily by chromosomal breakage or by aneuploidy. So this is what you can see under flow cytometry. Now from this video, you were able to understand about micronucleus assay and its importance, okay, and you were able to understand the the type of the damage it causes that can be used to identify. So thank you so much. Be curious. For any questions, you can always write mail to like, subscribe, and follow my YouTube channel, Toxicology on Board. All the best for your exam.